Keep your visions to the sound. Keep your ears to the ground. Let your mind pick up the guitar. I'm gonna be a fucking superstar.
We're Team Dendrimer of the Materials Science and Engineering Capstone class of 2013, and our design is a passivation layer for biomedical implants using a PAMAM Dendrimer film. Titanium implants are widely used for medical applications because titanium has high toughness and strength and good biocompatibility. But the problem with titanium implants is that they corrode, leaving metal ions in the body. Bianco et al. Did a study in 1996 with titanium implants in rabbits, and after a year they found almost twice the titanium in the surrounding tissues as they did in their control group. That is a serious problem. Now, titanium and its alloys naturally form an oxide coating that generally protects it from corrosion. But in vitro, even that oxide can degrade based on many factors. One important method is pitting corrosion from changing concentrations of chlorine and oxygen in the surrounding tissue. To create a diffusion barrier to block chlorine from reaching the implant surface and degrading the oxide, we decided to use dendrimers, which are these dense fractal polymers. Due to steric hindrance from their fractal branches, they have a dense outer layer with large cavities in the inner part of the molecule. We wanted to have a good understanding of how dendrimers were actually acting in solution, so we decided to make a computer simulation in MATLAB. The system we used was the bulk solution, the dissolved chlorine ions, the dendrimer monolayer, and the titanium oxide. Altogether, it was modeled by creating a 3D matrix with filled sites acting as either chlorine ions or densely branched dendrimer areas. When considering the ions inside the square lattice model dendrimer, the chlorine can hop in any direction where sites are available. The immediate sites in each direction and the secondary sites in the orthogonal directions were checked for openings and for dendrimer branches. This data is compiled to determine the relative hopping probabilities for each direction, and a new site was chosen for the ion. In a simulated bulk solution over thousands of time steps, the diffusion of chlorine ions into and through the solution was recorded. The results from modeling show that the chlorine ions get trapped within the dendrimer branches and do not reach the titanium oxide surface. Compared to the control model without a coating, the dendrimer layer greatly reduced the amount of corrosion-causing chlorine in the bulk material. We fabricated titanium samples for a prototype to test the effectiveness of the dendrimer monolayer. We sputtered 1 micron of titanium onto silicon wafers of 1 centimeter square. We oxidized some samples in a sputtering chamber with plasma oxidation, while we put others in the annealing furnace in the UMD Nanocenter Fab Lab to oxidize thermally. Samples were labeled and incubated in a Ringer solution for 5 days at 37 degrees Celsius. EDS analysis showed that after incubation, there was more chlorine on the dendrimer-coated samples. We believe this is because the chlorine ions get caught in the Dendrimer branches and remain even after washing. In addition, ICP-OES revealed that Dendrimer-coated samples released fewer titanium ions in the surrounding solution than our control, which supports our theory that the Dendrimer coating is preventing chlorine from reaching the oxide surface. These results uphold our initial hypothesis that a Dendrimer coating would decrease titanium corrosion.